Good afternoon, everyone. Myself Silan Kumar Mangalaut, Assistant Professor of Modern Institute of Pharmaceutical Science, Indore. Today we discuss about the topic in mutation and subject in pharmaceutical biotechnology. This index, definition, history and classifications. Definitions: a mutation. Generally, mutation is mute or mutations, changes in the genes. Mutation is the permanent alteration of the nucleotide sequence. Change in nucleotide sequence of the genome of an organism, extra chromosomal DNA or other genetic elements. Other genetic element में change होना is a mutation. A sudden change in physical or chemical properties of gene or gene makers. Physical and chemical properties of gene की physical chemical properties में changes. Mutation result from errors. It is a remove of the errors in DNA sequence, DNA replications, DNA transfer and DNA synthesis. It is damage of DNA, remove the damage of DNA. So mutation is contained the errors during DNA replication or the type of damage to DNA. This is history. The term mutation was originally coined by Dutch botanist Hugo de Vries. He discovered a new form of the evening primrose. It is discovered in evening primrose flower, scientific name Inothera lemaclena, that were growing in a meadow. This classification. Classification is a three bases. Such as first classify on the basis of cell in the plant and human cells. Classify on the basis of genetic inheritance. Classify on the basis of nature. First is a classify on basis on the cell. First, somatic. Vegetative mutations. Somatic is a condition in the non-gamete cell. It is not contain the reproductive cells. Other cells including the reproductive cells. Genetic alteration acquired by a cell that can be passed to the progeny of the mutated cell in the course of cell division. Second, gametes, germinal mutations. It is any detectable variations within the germ cells that when fully developed become sperm and ovum. It is completed a reproductive cell, gamete cells. Changes in the gamete cell such as the germinal mutations. Second, classify on the basis of genetic inheritance. Genetic inheritance is dependent on the chromosomes. First, chromosomal mutations. Second is a gene mutation. What is chromosomal mutation? Change in structure and number of chromosomes. Changes in the structure of chromosomes, number of chromosomes. Gene mutation. A gene mutation is a permanent alteration in the DNA sequence that makes up a gene. Changes gene में change होना, it is gene mutation. Chromosome में change होना in form of structure, in form of numbers, to show the chromosomal mutations. Both are different. First is chromosomal mutations is a two types: morphological, structural, and numerical. Morphological is a deletion, duplication, inversion, and translocations. Numerical is a euploidy and aneuploidy. First, deletion. Deletion is a morphological. Deletion occurs when nucleotides are left out of gene. मतलब when sequence is removed in the other DNA sequence. It is a deletion. Delete माना. It condition to delete. They also usually cause a shift in reading frame that will ultimately truncate the protein sequence shift करना. Deletion can be caused by errors in chromosomal crossover during meiosis, which causes several changes. During meiosis, the errors are founded, so they remove the errors by the deletion mutations. Duplication is the both sequence has very good form in the gene, so there is a duplications. A required of the duplication. A portion of the chromosome is duplicated, resulting in extra genetic material. Extra genetic material के द्वारा duplicate मतलब कोई सीक्वेंस में चेंजेस आना है, डुप्लीकेट हो जाना. डुप्लीकेशन अराइज फ्रॉम एन इवेंट टर्म्ड अनइक्वल क्रॉसिंग ओवर that occurs during meiosis between misaligned homologous chromosomes. It is condition to homologous chromosome. Inversion changes, frame shift changes. A portion of the chromosome has broken off, turned upside down, such as in this diagram A B C D E and F G H I J. So the inversion is GHI is inverted, IHG turned upside down and reattached. Therefore, the genetic material is inverted. Genetic material inverted is the term inversion mutations. or sequence changes. Translocation is sequence is other change. One type sequence is cut and paste. Chromosome translocation is caused by rearrangement of the part between non-homologous chromosome found in the non-homologous chromosome. Cut and paste. Other sides are cut and both different side is paste. Is a translocation. Changes. Position is changes in this chromosome, in these mutations. Such as around overall figures: deletion, insertion, inversion, translocations in the diagrams. Firstly, deletions, the insertions, inversions, it changes, or there are translocation, sequence changes. So the chromosomal mutations, a morphological chromosome mutations. Second is a numerical mutations. Numerical is a part of chromosomal mutations. Change in number of chromosome. Thus is two types is change in number of chromosome. So euploidy and aneuploidy. Euploidy number of chromosome changed by one set. One set is n, is a euploidy, such as haploid, diploid, triploid. Haploid is a one set of chromosome, is n. Diploid is a two set of chromosome, 2n. Triploid is a three set of chromosome, 3n. Polyploid is a more than chromosome, more than three set of chromosomes. Aneuploidy, number of chromosomes changed by chromatid, is a aneuploidy. Less than minus is a monosomy, -1. Nucleosomy, -2. More than plus trisomy, +1. Tetrasomy, +2. is a aneuploidy. Number is changes. The chromosome is a minus is a monosomy, nucleosomy. Plus 1 is a trisomy, tetrasomy. It is a numerical mutations. Second is a gene mutation. Gene mutation is a permanent alteration in the gene sequence of the nucleotides. Gene mutation such as point mutation, inversion mutation, frame shift mutation is a types of gene mutations. Three types. Point mutation is a three types: same sense, antisense, missense, or nonsense. Such as point mutation. Point mutation is a mutation that only affects a single nucleotide of nucleic acid. Only affect a single nucleotide of nucleic acids. Point mutation most commonly involve the substitution of one base for another. One base for another is a point mutation. Only point sequence, एक point पे change होना is a point mutation. Which change the complementary base as well in DNA, such as sickle cell anemia. Such as the genetic code GAG. GAG changes the AUG. GAG is a glutamic acid and GUG is a valine. Only change in the one sequence. एक form is U. एक ही जगह पे U का changes होना, it change in the amino acids. Is a point mutations. Same sense is a one sense with changes such as point mutation के types में है same sense, मतलब one sequence change होना. जैसे एक GAG is a glutamic acid, लेकिन GCG is a glutamic acid. Both are same. It is a one sequence in change. In replaced by the C, but glutamic acid of same. Amino acid is not change. It is same sense. Antisense is the changes the amino acids. GAG glutamic acid, GUG valine. It changes the amino acids. Nonsense is the changes the genetic code, but amino acid is not required. Such as GAG glutamic acid, but changes the G in the form of U. UAG is a nonsense codon. It is not is a amino acids. UAG किसी भी फॉर्म में कोई जेनेटिक कोड नहीं है, इसलिए से नॉनसेंस कोड बोलेंगे. इसलिए से नॉनसेंस बोलते हैं कि अमीनो एसिड चेंजेस नहीं हो पाते. इट इज अ पॉइंट म्यूटेशंस. सेकंड, इन्वर्जन म्यूटेशन. एंड इन्वर्जन इज a genetic code reversed in which a segment. Inversion is such as GAG genetic code. GAG is a glutamic acid, but changes the inversion. AG is change in the GA. So the GGA is a glycine. It is a inversion mutation. मतलब change the sequence is 1 2 2 1 is a sequence in form of the sequence changes. So the amino acid is change. Third is a frame shift mutation. Frame shift mutation, also called a framing error or a reading frame shift, is genetic mutation caused by indels, insertion or deletions of a number of nucleotides in a DNA sequence. Is a frame shift mutation, such as the red color is a GC is delete, and last form is GC is addition. Deletion, addition is a frame shift mutation. Only frame are changed. It is a frame shift mutations. Such as in the diagram, the codon changes the C in form of the C. It is a frame shift mutations. Only one changes. Nucleotide sequence changes. It is second type. Third type classify on the basis of nature. Third classification of the mutations. Nature in the basis of nature. First is spontaneous mutation. Second is induced mutations. Spontaneous mutation is certain mutations. It depend on nature. Nature is the environments, whether that of plants, animal sources, motions. It is changes. The spontaneous mutations are result of errors in natural biological process. Natural biological process changes the spontaneous mutations. Induced mutation provide the induced. Induced mutations are due to agents, mutants in the environment that cause changes in DNA structure. Mutagens is the chemical substance. Those are changes in the mutations such as electronic rays, X-rays or gamma rays, ultraviolet rays, and such as chemicals, fragrance, synthetic chemicals are changes the mutations. It is a induced mutations, such as change in the DNA structures. Thank you.